Welcome to the TOEFL listening section. In this part, you will hear one or more conversations in an academic or campus context, such as a discussion between a student and a professor, or between two students. You may take notes while listening. Listen to part of a conversation. You will hear it only once, so listen carefully. After the conversation, you will answer questions about what you heard. Some questions will ask you about main ideas and details. Others will ask about implied meaning, speaker attitude, or how ideas relate to one another. Use both what is stated and what is implied to answer.
Good afternoon, Dr. Morris. Thank you for meeting with me during your office hours.
Hi, Jason. No problem. You mentioned you wanted to discuss your project on targeted drug delivery using nanoparticles.
Yes. I am trying to refine my research question. I am particularly interested in how nanoparticle design parameters influence tumor targeting and release kinetics.
That is a good focus. Material science plays a central role here. Size, shape, surface chemistry, and mechanical properties all change how nanoparticles behave in biological systems.
For size, I read that particles between 20 and 180 nanometers exploit the enhanced permeability and retention effect in tumors. Is that the right range to aim for?
Yes, that is the common guideline. Particles below 20 nanometers are cleared rapidly by renal filtration. Particles larger than about 200 nanometers are taken up more by the spleen and liver. Staying in that middle window increases passive accumulation in tumor interstitium through the enhanced permeability and retention effect.
I see. But passive targeting seems limited by variability in tumor vasculature. How do we improve selectivity for cancer cells?
You can add active targeting. If we conjugate a ligand that recognizes a receptor overexpressed on tumor cells, we can achieve active targeting and increase internalization by endocytosis.
Could you give me examples of ligands used for active targeting?
Common ligands include monoclonal antibodies, antibody fragments, peptides such as RGD that bind integrins, and small molecules like folic acid that target folate receptors.
So for breast cancer, I might conjugate an antibody fragment that binds HER2 receptors. But does adding antibodies change circulation time or cause immune reactions?
It can. Antibodies increase molecular weight and may enhance recognition by the mononuclear phagocyte system. To mitigate that, we often use polyethylene glycol on the surface in a process called pegylation to reduce protein adsorption and improve circulation time.
Right. I have seen pegylated formulations in clinical use. For instance, Doxil is a liposomal doxorubicin that is pegylated. Are there other clinically approved nanoparticle drugs I could cite in my literature review?
Yes. Abraxane is an albumin-bound formulation of paclitaxel, and Onpattro is a lipid nanoparticle used for siRNA delivery. These are good case studies to show translation from materials design to clinical application.
I want to compare liposomes to polymeric nanoparticles in terms of drug release control. Which materials give better tunability of release kinetics?
Polymers like PLGA allow more precise control of degradation-based release. You can tune monomer ratio and molecular weight to vary release over days to weeks. Liposomes are more immediate, but you can modify their bilayer composition to slow release. Stimuli-responsive systems add another layer of control.
When you say stimuli-responsive systems, what stimuli are commonly used in tumor targeting?
Tumor microenvironments often have lower pH, elevated enzyme activity, and sometimes higher temperature. Materials that respond to acidic pH or matrix metalloproteinases release payloads selectively in the tumor. Magnetic nanoparticles can be activated by external magnetic fields, and light-responsive nanomaterials use photothermal heating.
Photothermal therapies with gold nanoparticles sound promising. Are there examples where gold is used for both imaging and therapy?
Yes. Gold nanoshells and nanorods are used as theranostic agents. They absorb near-infrared light and convert it to heat while also providing contrast for imaging. That dual function is attractive from a materials perspective.
What are the main characterization techniques I should mention for my methods section to show how we evaluate nanoparticles?
Dynamic light scattering is used for hydrodynamic size and polydispersity. Transmission electron microscopy provides particle morphology. Zeta potential measurements show surface charge. Drug loading and release are quantified by chromatography and mass spectrometry.
I worry about toxicity and long-term clearance. How do materials scientists address nanotoxicology?
You must evaluate biodistribution, acute toxicity, and immunogenicity in relevant models. Surface chemistry and biodegradability are key. Using biodegradable polymers reduces long-term accumulation. We also analyze protein corona formation to understand how the biological identity differs from the synthetic identity.
Protein corona formation complicates targeting because it can mask ligands right.
Exactly. That is why surface engineering and predictive in vitro assays are important. Designing stealth surfaces and testing in serum conditions helps anticipate in vivo behavior.
From a translational standpoint, what are the biggest hurdles to bringing a nanoparticle drug to clinical use?
Regulatory requirements for reproducible manufacturing, scale-up, batch-to-batch consistency, and thorough safety profiling are major hurdles. Also demonstrating a clear therapeutic advantage over existing treatments in clinical trials is essential.
That makes sense. For my project, I plan to prototype polymeric nanoparticles with a targeting peptide, pegylation for circulation, and an enzyme-responsive linker for release.
That is a solid plan. Make sure you include characterization steps and controls to assess targeting specificity and release in relevant cell models and an animal model if you can.
Thank you. One last question. How important is particle shape compared to size and surface chemistry?
Shape matters. Rod-shaped particles have different margination and cellular uptake profiles than spheres. Disks and filaments circulate differently. Shape interacts with size and surface features to influence biodistribution and cellular internalization.
Great. I will incorporate these considerations and prepare a revised proposal and a timeline. Thank you for the guidance.
You are welcome. Send me your draft and we will discuss experimental design and key assays next week.
Listen again to part of the conversation, then answer the question.
If we conjugate a ligand that recognizes a receptor overexpressed on tumor cells, we can achieve active targeting and increase internalization by endocytosis.
This concludes the conversation portion. For more TOEFL practice tests and study materials, visit Spinta.org.